This section on muscles is about the sliding filament theory. So we have to talk about these filaments. So remember, the main proteins are actin and myosin. Actin is thin and myosin is thick. However, we have these extra proteins, and you'll see 4 of them here. Well, actually 5 of them here. Actinin, F-actin, nebulin. But the only ones we're concerned about is the tropomyosin and the troponin. So you'll see all these different proteins in here. This whole strand here is the actin. While it looks very thick because of all the protein globs here, it is the actin, the thin actin. So here's the tropomyosin. The tropomyosin kind of wraps around the actin, and what it does is it blocks these binding sites. And when the binding sites are blocked, the myosin cannot attach to the actin and pull it. One thing that is going to unblock the tropomyosin from the actin is troponin. And once calcium binds to troponin, it moves the tropomyosin out of the way. So here's our thick filament, the myosin. Again, too many proteins. We don't care about the titin. But you will see that the myosin consists of what are called heads. These are the myosin heads. So the tail faces the M line, which is the center, and the free heads face outward toward the thin filaments. So here's our sarcomere. The sarcomere runs from the Z line to the Z line. Attached to the Z line are the thin actin filaments in pink, and the thick actin filaments are in between, the thick myosin filaments are in between the actin filaments. So here's a close-up of our thick myosin with their myosin heads. Well, with the sliding filament theory of muscle contraction, the filaments slide past each other when the muscles contract. So if you look at the width of this sarcomere at rest here on the left, when muscles contract, the Z lines move closer towards each other, the filaments slide past each other, some of these bands shorten up, and the muscle contracts. So if you were to look at individual bands, you would see that the H band gets smaller, the I band gets smaller, the zone of overlap gets bigger, those Z lines get closer together. With the sliding filament theory, every single sarcomere gets smaller. So if your muscle were not attached, such as this muscle filament down here, were not attached to any bone, the entire filament would get smaller, and you'd see it decreasing at each end. But keep in mind, our muscles are attached to a bone, so if you had this end here on the left attached, it would get shorter from just the single end. The next section is about membrane excitability. All the membranes in our body have ions on both sides of them. And these ions can cross the plasma membrane and change the positive and negative ions on each side of the plasma membrane. So with these ions on both sides of the membrane, usually the inside of the membrane is more negative than the outside. So inside the membrane is negative, outside is positive, and this is called polarized. There's a difference in those, in the distribution of those ions, and that difference is called the membrane potential. And this membrane potential is measured in millivolts. Neurons have resting membrane potential of negative 70 millivolts, and skeletal muscle fibers of negative 85 millivolts. So here's a picture of that. This is the plasma membrane. Inside the plasma membrane are negative charges. Outside the plasma membrane are positive charges. And this is just showing that some of the things that cause the negative charges on the inside are these proteins, which do not travel across the membrane. So you always have these negative proteins inside the membrane. So as you know, these plasma membranes are selectively permeable. The outside will be different from the inside because the cell has selected what to bring into the cell. Outside the cell has more sodium. So think of our cells as being in the salt water. There's more sodium outside. Inside the cell prefers more potassium. Those are both positive charges. The negative charges are proteins in the cell, and they're going to stay in the cell. And there's a lot of chlorine outside because remember, we are in a sodium chloride or salt water sea within our body. The constant sodium outside the cell, well, this is a problem for all living organisms, that the salt tends to flow into the cell. These are through what are called leak channels, and so sodium enters the cell because there's more sodium outside, and it kind of diffuses into the cell. The potassium diffuses out of the cell through a leak channel, and this is just the natural state of the membrane. But the cell does not accept this, and so a sodium-potassium pump system has developed so that, yes, even though sodium constantly diffuses into the cell, the pump will pump it out. And yes, even though potassium always diffuses out of the cell, the pump will pump potassium back into the cell. So the pump works this way. For every 3 sodium pumped out of the cell, there will be 2 potassium pumped into the cell. And this requires ATP. So an action potential, we have to understand the action potential and how that occurs on the muscle membrane. So let's begin with a normal cell, negative inside the cell, positive outside the cell. Well, there will be a small increase in sodium ions entering the cell. So there are positive ions entering the cell, making inside the cell less negative. So it's moving the inside charges in a more positive direction. This causes voltage-gated channels to open, and a lot of sodium rushes into the cell, and this results in a depolarization. There's been a change of the membrane, so it's positive inside the membrane and negative outside the membrane. Those sodium charges result in that change in positive charges inside the membrane. So the membrane potential reaches plus 30. Then those voltage-gated sodium channels close and voltage-gated potassium channels open, and potassium leaves the cell. So remember, inside the cell, it was too positive because the sodium had entered. So these potassium channels open up and potassium leaves the cell, so the charges return to normal. The ions are not normal. The cell wants potassium, but potassium is leaving the cell. The membrane potential returns to normal, and this is called repolarization. Once resting potential is reached, that is, the cell has returned to pre-action potential state, those voltage-gated potassium channels will close, and now there's too much sodium in the cell and too little potassium, so the sodium-potassium pump returns the ions to the correct resting potential ions. The refractory period is the time needed to reach the original distribution of these ions, and the membrane will not respond to a stimulus until those ions have been restored to normal. So pay attention to this figure, small increase in ion permeability. And so remember, the membrane starts at -70 millivolts. It's just a measure of how different the membrane is from inside to outside. There's a small increase in sodium permeability. Sodium enters the cell, and inside the cell it becomes less positive, -55. This causes the voltage-gated sodium channels to open, so sodium rushes in very, very quickly. And this results in depolarization of the cell. That is, there's a switch in the membrane potential, and the cell becomes positive inside and negative outside. Once that happens, the voltage-gated channels close and the voltage-gated potassium channels open. Potassium is drained from the cell so that the cell returns back to the original resting potential. Those voltage-gated potassium channels close, but the membrane situation is not right in terms of the ions, so the sodium-potassium pump has to act to pump out 3 sodium ions for every 2 potassium ions coming into the cell. And during this refractory period, the cell will not accept another action potential.